This module is on interpretation of the hemifields. For hemifield recordings, typically 5 electrodes are attached at the back of the head, and these are the active electrodes, and they are referenced to FZ. Typically, the placement is according to the Queen square method, as opposed to the 10-20 method. And so the midline occipital electrode is placed at 5 centimeters above the inion. 2 left hemisphere electrodes are placed. 5 centimeters left of the mid-occipital electrode is the left occipital electrode, and then 5 centimeters to the left of that is the left temporal electrode. And in the same way, a right occipital electrode and a right temporal electrode are placed. This is an illustration of the normal VEP response to hemifield stimulation. What you'll note here is that there are 4 tracings on the right-hand side that show the N70 P100 VEP response at 4 of the recording electrodes. The 5th recording electrode, the right temporal electrode, is not shown in this illustration. So we're going to start at the left side of this picture where we see the checkerboard, and you'll note that the checkerboard is on the right visual field. It's on the right side, and so it's a right visual field stimulation. Stimulations of the visual fields cross over to activate the opposite retinal fields. And so this right visual field stimulation activates the left retinal field of the right eye. And so in this picture, we see that the right eye is the one being activated, and it's the left retinal field of the right eye that's receiving the right visual field stimulation. This left retinal field of the right eye is also called the nasal field of the right eye. An activation of nasal fields cross over at the optic chiasm. So in this case, we have a right visual field checkerboard activating the left retinal field of the right eye, and that activation travels down the right optic nerve and crosses over at the optic chiasm. This then passes into the left hemisphere and travels to the left visual cortex. And as you know, responses in the left visual cortex are actually seen as larger responses over the right recording electrodes because of the infolding of the visual cortex. And so in this example, we have activation of the left occipital cortex showing a clear N70 P100 at the mid-occipital and right occipital electrodes. And what you'll also note is that there's a phase reversal between the right occipital electrode and the left temporal electrode, where the N70 and P100 are phase reversed with each other. And so this is a very normal response because they cross the cortex. The cortex where the response is generated creates a dipolar response, and so we see the typical VEP in the right hemisphere, and in the left hemisphere we see its phase reversal. So for a hemifield recording, we systematically stimulate each of the hemifields in each of the eyes. And by looking at the pattern of VEP responses with each of the hemifield stimulations, we can start to localize lesions within the visual system. And so we'll talk about that on the following slides. When we talk about hemifield interpretation, we often see a figure that looks like this, which summarizes the different defects that can happen in the visual pathway and the pattern of results that would be observed. And so we're going to walk through this figure slowly. What I want to point out is that we'll see there's a left eye and a right eye, and for each eye we see the visual field that's stimulated. So the red is always the left visual field, and so there's a left visual field in the left eye and a left visual field in the right eye, and they both activate the right retinal fields in the eye, which in the left eye is a nasal field and in the right eye is a temporal field. Okay, and then so we can follow the pathway by using the colors to see where the pathways end up. Now the piece on the right that says visual field with a bunch of circles of different colors, what this represents are the visual fields that can be obtained. And so, for example, for number 1, the gray part means that the visual fields are not seen. And then if we see the blue and red, it means that those visual fields are seen. And so you might understand a little bit better on the next slide. So on this slide, we see that there's a black box labeled number 1 that cuts the left optic nerve. And so what this means is that there's a lesion in the left optic nerve, and so the stimulus cannot pass through the left optic nerve. And so that you'll see on the visual field drawing on the right, there's nothing coming in through the left eye, and so it's colored gray. Whereas for the right eye, both the red and the blue come through, and so both the right and the left visual fields are able to travel down the optic nerve in the right eye. And so this is a monocular lesion. It's a lesion of the left eye. This is an illustration of what is observed when we have a lesion in one eye. And so this is a full field stimulation. It's just a little bit different from what we were just looking at, but it doesn't matter because it's the entire optic nerve of one eye that is lesioned. And so starting at the top, you'll see the full field checkerboard activates the right eye, and it activates the entire retinal field of the right eye. And so we see a normal looking VEP response because the right optic nerve is intact. When we move down to the bottom illustration, we see the same stimulation activating the entire retinal field of the left eye. However, there's a lesion in the left optic nerve, and so the stimulus cannot pass by this lesion, and so there is no input going to the cortex, and so you'll see that the tracings are flat. So in this figure, we're looking at a lesion at number 2. So it's cutting the blue input on the left eye. And so this is the temporal input of the left eye that comes from the left retinal field. And so what this means is that any input that comes to the left retinal field, the blue side of the left eyeball, cannot pass through to the brain, whereas everything else will pass through to the brain. And so you see on the right-hand side that the gray part, so the right visual field, which activates the left retinal field, is gray because nothing can pass through on the left temporal field of the left eye. Whereas input to the left visual field of the left eye can activate the right retinal field of the left eye, and so we see the red of the left eye. And then also on the right eye, there's no lesion anywhere, and so we see the full red and blue stimulation going through. Lesions of either temporal fields of either eye will show the same pattern of results, except that in one eye it'll be on the left, and in the other eye it'll be on the right. This image shows a lesion at number 3, and so there's a lesion at the optic chiasm. And so when there's a lesion at the optic chiasm, the fibers can't cross over. And so starting at the left eye, the blue fibers, the temporal fibers, stay ipsilateral, and so they will continue and can be seen. Whereas the red fibers of the left eye, the nasal fibers, are the ones that cross over, and so they cannot cross over because there's a lesion at the optic chiasm. Now for the right eye, the nasal fibers are the ones that cross over, and so they can't cross over because of the lesion at the optic chiasm, whereas the temporal retinal fibers of the right eye stay ipsilateral in the right hemisphere, and so they continue on. And so we see this pattern on the right. We see this pattern on the right figure, labeled number 3, where both nasal fields, the inputs for both nasal fields can continue on and are observable. So we see the blue and red, whereas both temporal fields, the temporal fields of both eyes, cannot cross over. Their input cannot go on because of the cut at the optic chiasm, and so they are colored gray. Continuing on, we see in this figure a lesion at number 4, which is a lesion at the left optic tract. And so with this lesion, which is post-chiasmal, Anything that goes through the left optic tract cannot continue on to the left brain. And so following the blue colors, we'll note that the left optic tract is lesioned, and that receives input from the left retinal field of both eyes, which corresponds to the right visual field of both eyes. And so all of the blue, the left retinal fields and the right visual fields, are not passed on to the left optic tract. And so when we look at the picture on the right, corresponding to number 4, what we see is that the red input, the inputs that come from the left visual field going to the right retinal fields of both eyes, will continue down the right optic tract, and so they are able to generate a VEP, whereas the blue parts, the right visual field, which activates the left retinal field and the information is translated to the left optic tract, cannot pass on to generate a VEP, and so those parts of the visual field are colored gray. And so finally, here we see a lesion at number 5, which is at the left optic radiations. And you'll note that number 4 and number 5 look very similar. So lesions at the optic tract and lesions at the optic radiations cannot be differentiated by looking at the pattern of the VEP results. This is a chart that summarizes the patterns of abnormalities of the VEP and what they would localize to if we saw this pattern of abnormality. And so starting at the top, if we saw a binocular absence of VEP, so in both eyes there is no VEP recorded, some of the things we need to think about are perhaps there's a technical problem and the stimulus is not getting through, or the recording is not being made, and so we need to consider technical problems first. Next, if there are no technical problems, then we need to consider an eye problem. And so perhaps the absence of a VEP from stimulating both eyes is due to a problem in either or both eyeballs, an ocular problem. And then third, a binocular absence of the VEP might suggest bilateral optic nerve defects. Moving down to line 2, if there is a monocular absence of the VEP, then this may correspond to an optic neuropathy or an optic nerve lesion in the eye that did not generate a VEP. But what also needs to be ruled out is that there isn't an ocular problem in that eye. Moving down to line 3, if an abnormal VEP is seen to temporal stimulation of the bilateral visual fields, so we know that the bilateral temporal visual fields cross over and activate the nasal fields. And so the nasal fields are the ones that cross over at the optic chiasm. And so if we see that there are abnormal VEPs, when we stimulate the temporal visual fields, then we know that it's a nasal retinal field problem, and so the lesion would be at the optic chiasm. Moving down to line 4, if there's a lesion post-chiasmally at the optic tracts or at the optic radiations, that would generate an abnormal VEP in the contralateral recording electrode. So for example, if there is a lesion at the left optic tract or the left optic radiation, this would show up as an abnormal VEP at the right recording electrodes. And this left optic tract or left optic radiations are receiving inputs from the left retinal fields of either eye, and so this would correspond to stimulation of the right visual fields. And so if we stimulate the right visual fields of either eye and note an abnormal VEP, then we note an abnormal VEP at the right recording electrodes, then we know that there is a lesion in the left post-chiasmal tracts. And the same idea if it were the right optic tracts or the right optic radiations. This pattern of abnormalities of the VEP would suggest a homonymous hemianopsia. This last line corresponds to the number 2 cut that we saw in the previous drawing. And so if there is an incomplete lesion of the optic nerve, what would be seen at the VEP is quite specific. And so first it would be specific to the eye that's affected, and then there would be an asymmetry in the lateral VEPs where the side that is affected would correspond to the side of the optic nerve that is lesioned.